next we'll discuss about anaphase in mitosis. So now in the last session we discussed about metaphase. Like in metaphase, all chromosomes, they were arranged at equator, right? So now what happens that those chromosomes, they will split simultaneously, and two daughter chromatids, they will be now called daughter chromosomes of future daughter nuclei. So here migration will take place, migration towards the opposite poles. Like let's say this was the position in metaphase. Okay, now in anaphase, since spindle fibers are there, now what will happen, this chromatid, this will pull towards itself. So chromosome will move away from the equatorial plate, and the centromere, that will remain directed towards the pole. It will be like this. This will, in anaphase, they will migrate towards the pole. And in this side, this chromatid will come here. It will pull towards itself, these spindle fibers. So centromere along with centromere, so this centromere will split. One will go to one pole and the other one will go to second pole. So centromere here, centromere will split. Then chromatids, this is one chromatid here, other chromatid here. So chromatids, they will get separate and they will move to opposite poles. Okay, so this was about anaphase. So if I draw the anaphase, it will look like this. This is the centriole, and these are the spindle fibers. This is the splitted centromere. This is basically metaphase, and this is the anaphase. Okay, now coming to after anaphase, we have telophase. So this is the final stage of karyokinesis. 4 stages of karyokinesis were there: prophase, metaphase, anaphase, telophase. So this is the last and final stage of karyokinesis. So the chromosomes now, they have reached their respect. So if you see the last condition, right, chromosomes, they are here at opposite poles. These are the microtubules, chromosomes like this. So chromosomes, they will move towards opposite poles, and here they will lose their individuality. Individuality means they will split into two, one chromatid upside and one chromatid downside. So the individual chromosomes can no longer be seen, and each set of chromatin material that will be placed at two poles. Now here what happens, telophase, it will be like this. This is about anaphase. This is the centriole. So nuclear envelope develops around the chromosomes cluster. Wait, it will be like this. Nuclear envelope develops around the chromosome clusters at each pole, and now these are two daughter nuclei will be formed. Daughter nuclei. This is what basically karyokinesis is, nuclear division. Here nucleus is also getting divided. First centromeres were splitted, now nucleus is also getting divided. This is basically what karyokinesis, final stage of karyokinesis. So now here the cell organelles that will reform, like in prophase no cell organelles were formed, but now this is the last and final stage. So all cell organelles, nucleolus, Golgi complex, then endoplasmic reticulum, they will form, they will reorganize, they will reform. So here what happens? Nuclear envelope develops around the chromosome like this. Then cell organelles like nucleolus, Golgi complex, endoplasmic reticulum, they will reform, they will reorganize, all are same terms. Okay, then there is there is cytokinesis. After karyokinesis comes cytokinesis. Cytokinesis means like the last stage in telophase, it was like this. Now what happens in cytokinesis? There is division of cytoplasm. It will form like this, separate daughter cells. So now here we have the, like this, here we have the chromosome, that is the parent cell. So here cell is divided into two daughter cells by the separation of cytoplasm, which is called cytokinesis. So this completes the cell division. Now there are some other key points, like in an animal cell. Now if I talk about animal cell, now how this is in animal cell cytokinesis, this is achieved by appearance of a furrow in the plasma membrane. Furrow means like this. There is a furrow in the plasma membrane, then the furrow gradually deepens and ultimately joins in the center, and it will divide the cell cytoplasm into two. Okay, then plant cells, they undergo cytokinesis. Now how? By inextensible cell wall. That they are in plant cells, they are enclosed by inextensible cell wall. In animals, it is done by furrow cytokinesis. In plants, this is done by cell wall. Okay, and now here's a different mechanism in plant cells. What happens? Wall formation starts in the center of the cell. This is the cell. Wall formation starts, and it will grow outward to meet the existing lateral outward. It will grow outward. Okay, then the there will be the formation of new cell wall, and that forms the cell plate. Further, it will form that cell wall will form the cell plate. So basically that is middle lamella between walls of two adjacent cells. So basically animals and plants, this is not that important. This is only important. Two daughter cells are formed. So basically at the time of cytoplasmic division, organelles, they like mitochondria and plastids, they also get distributed. Since two daughter cells are formed, all organelles will be formed, like mitochondria will be formed, then plastid will be formed. So now there is another point that, let's say in some organisms, says what happens, karyokinesis is not followed by cytokinesis. The karyokinesis is there, but cytokinesis is not there. So what happens? There is a multinucleate condition. Multinucleate condition is there, which leads to the formation of syncytium. For example, liquid endosperm in coconut. Okay, so this completes the cell division. Okay, so how it was like, if I just see the overall performance, it was like G1, then it was S, then it was G2, undivided stage, that quiescent stage, that is G0, then it was M phase, and M phase that was further divided into 4 stages: prophase, metaphase, anaphase, telophase. This all constitutes the karyokinesis. After karyokinesis, there comes cytokinesis. Okay, so in G1 what happens? Cell will just actively divide, no replication will be there. In S, replication will be there. Here replication will take place. Here it will grow continuously, no replication. Here G2, proteins will be synthesized. Prophase, 2 phases are there, early and late, condensed form and untangled form. In metaphase, they are aligned at the equator. In anaphase, they will get separated. Okay. In telophase, it is like this final stage of karyokinesis, that is nucleus will divide here. Okay, and at last what happens? Cytoplasm. Cytoplasm is this is the cytoplasm. Okay, this pink color, this all is at the end stage cytokinesis, cytoplasm will divide. So it is like this is how mitosis takes place, and it takes place in diploid cells, not in haploid cells. Diploid means like the normal cells of our body, liver cells, kidney cells, heart cells, these all are diploid cells. Okay, so this was about it. Thank you.